Hello everyone, it's my pleasure to present our latest work in this FAST Foundation Symposium. Today, I'm going to talk about a study investigating the feasibility of non-invasive brain mild hyperthermia using transcranial MRI-guided focused ultrasound in rodents. Around 700,000 people in the United States are living with a primary brain tumor. 80,000 more will be diagnosed in 2020. Current treatment options include surgery, radiotherapy, chemotherapy, and the emerging immunotherapy. However, for certain brain malignancies, for example glioblastoma, as shown in the figure, the overall survival and 5-year survival rate remain low with the current standard of care. Therefore, novel therapies or techniques are highly desired. Mild hyperthermia can directly induce tumor cell death, can promote chemotherapy and radiation therapy by enhancing localized drug delivery and sensitizing tumor cells to radiation. It also has the potential to activate the immune system to augment immunotherapies. So the goal of this study was to investigate whether it is feasible to achieve focal brain mild hyperthermia using transcranial MRI-guided focused ultrasound. We also investigated if microbubbles can drop the ultrasound power during hyperthermia, thereby reducing skull heating and improving the safety. The system we used is a small animal FAST system purchased from Image-Guided Therapy in France. As the bottom inset shows, a transducer of 2.5 MHz was placed on top of the animal head. The study was done in a 9.4 Tesla Broca MR scanner. During hyperthermia, either saline or microbubble solution was infused into the animal through the tail vein. The left is the actual MRI image showing the transducer coupling water and ultrasound gel and the mouse brain. We found that a water phantom with a temperature sensor embedded, placed right next to the animal head, was essential for phase drift correction for accurate MR thermometry. The right figure shows the FAST target region overlapped on an anatomical brain MRI image, and the drift RI selected in the water phantom. The video here shows a representative 5 minutes hyperthermia experiment with 5 minutes cooling. Definity was infused into the animal during hyperthermia. The right figure shows the ultrasound power in purple and the target temperatures as a function of time. The mean temperature in the target region, as shown in the green line, indicate the temperature was well controlled at 40 to 43 degrees. When comparing the saline group with the microbubble group, we found that both thermal dose CM43 and the ultrasound power reduced with microbubbles in 10-minute or 5-minute hyperthermia experiments. As marked by the dashed circles, in the FAST target region, no edema, hemorrhage, or blood-brain barrier permeability change were observed in both saline and microbubble groups, as shown in T2-weighted, T2 star-weighted, and the contrast-enhanced T1-weighted MRI images. However, we do realize that in the superficial cortical regions under the skull along the FAST beam path, there are signs of skull heating associated damage. This was confirmed by histology. If we compare saline and microbubble groups, especially when we look at the figure B and figure C, we can see microbubbles reduced a lot of this type of damage. In conclusion, it is feasible to perform focal brain mild hyperthermia using transcranial MRI-guided focused ultrasound. There was no evident damage or blood-brain barrier permeability change within the FAST target region, with or without microbubbles. The presence of microbubbles showed evidence of reducing ultrasound power required for brain mild hyperthermia, therefore reducing skull heating associated damage. In future, we will investigate microbubbles with higher bubble concentrations and transducers with lower ultrasound frequencies to see whether we can totally avoid this skull heating associated damage. I would like to acknowledge these funding resources to support this study. Especially, I want to thank the Alberta Innovates for granting me the postdoc award in health innovation with generous financial support. Thank you for attending.